Hello, we're glad you've joined us for this live webinar, When Confidence and Consciousness Collide, Neural and Computational Approaches to Understanding How the Brain Creates Subjective Experience. I am Christina Mahalik of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. To learn more, visit labroots.com. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, just simply type them in the Q&A box, which will open when you click on the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience any technical problem seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report the problem by clicking on the green Q&A button at the lower left of the screen. It is now my pleasure to present today's speaker, Megan Peters, PhD, postdoctoral researcher, University of California, Los Angeles. Megan Peters is a postdoctoral researcher at the University of California, Los Angeles. She received her BA in Cognitive Science from Brown University in 2006 and her PhD in Computational Cognitive Neuroscience from UCLA in 2014. During her doctoral training, she received a National Science Foundation Graduate Research Fellowship and a National Institutes of Health Training Fellowship. Megan's research uses computational models and machine learning approaches to explore the neural computations underlying perception, metacognition, and subjective awareness. Her research uses a multidisciplinary approach to gain insights from the human behavioral and neuroscience, as well as non-human primate and rodent models. Dr. Peters will now begin her presentation.
Okay, thank you very much to LabRoots for hosting this virtual conference and for inviting me to share my research with you all today. As Christina just said, my name is Megan Peters. I'm a postdoctoral researcher at UCLA in the psychology department. And today I'm really excited to tell you about some of the research that I've been working on for about the past 2.5 years on the relationship between metacognition or confidence judgments and consciousness or subjective awareness itself. Okay, so for my entire scientific career, I have wanted to study consciousness. I want to ask, how do our brains create this subjective sense of the world around us, and what neural computations underlie the subjective nature of our perceptions and our metacognitive ability to reflect on those perceptions. And in my graduate work during my PhD, I studied perception itself, and I used computational models, so mathematical modeling, to describe the relationship between objective reality and subjective experience, especially when they don't match, as in the case of illusions. So a few years ago, when I finished up my graduate work, I felt I was in a great position to use computational models to study subjective consciousness. So based on my ideal observer modeling background, I wanted to show that visual masking breaks down optimality by creating something that looks like blind sight in normal observers. So the neurological condition of blind sight occurs in some patients who have damage to their primary visual cortex. This is the first cortical stop along the visual processing stream. And these patients can do some visual tasks, like correctly orienting a letter to put it through a mail slot that's vertical or horizontal, or telling you whether a line on a computer screen is tilted left or right. But they report no visual experience. They say they can't see, even though they can do these tasks. So from a scientific perspective, this disorder is really fascinating because it decouples the ability to see from the subjective experience of seeing. But this is really hard to study, of course, because these patients are very rare. So some researchers have tried to induce this effect where you have task performance, but you have no subjective awareness. They've tried to induce this effect in neurologically intact observers using techniques such as visual masking. So in visual masking, you make the stimulus very dim and you mask it with other images that are flashed very near it in space and time. And when you do this, even neurologically intact observers show similar behavior. They're able to do some discrimination task, but they report that they can't see the stimulus at all, that they cannot introspect on their choices, and they have low confidence in them. Okay. And so for me, this was really exciting to study. Okay, so there's this problem, though, when I looked at most paradigms that are used to study this phenomenon. Most of the time when people do this, they use a paradigm that looks like this. They show people some masked stimulus, and they ask for a decision about it, and also ask for some sort of confidence or visibility rating afterwards. So they assume that if there's high signal, you're going to say you saw the thing, and if there's no signal, you'll say you did not see it. And we'll call these the conscious and the unconscious trials, respectively. And so you demonstrate that on those unconscious trials, people can do the task, and then you've demonstrated suboptimality. But here's the problem. There's a third option, which is that the subjective signal is weak, but it's still there. It's just not strong enough to pass some sort of internal criterion or threshold to prompt the person to say, I saw the thing. So they say they didn't see it, even though they sort of did, but they can still do the task. Well, that's actually not very interesting, is it? There is a signal there, so of course they can do the task. They're just reluctant to tell you they saw the stimulus because the signal wasn't strong enough to pass some sort of internal criterion. And so this is the criterion problem, and it's really a problem indeed. So depending on where each observer puts their own internal line for dividing samples into seen and unseen reports, you might get subjects who see everything. They never use the bottom of the scale. They never say, I didn't see it, or I have low confidence. But importantly, you might also get subjects who see very little. They put that internal line too high, so they're really unwilling to use the top of the scale. And suddenly you think you've found blind sight in this normal observer, but all you've really found is someone who's not willing to report that he saw something. And this is really a concern for studies of consciousness, because when we see in an experiment that people can do some task but they report no awareness, that might not actually tell us anything interesting at all. So if we go looking for brain areas or other neural correlates that do this kind of unconscious perception, we don't even know what we're actually looking at. And this problem is far from new. People have been reporting it for 50 years, and it still exists despite previous efforts to solve this problem in the context of studying unconscious perception in this way. So clearly, if we want to figure out how blind sight indicates suboptimality in subjective or introspective computations, we're going to have to fix this. So we have to design a criterion-free task in order to be able to really conclude that this blind sight-like behavior with a masked stimulus is a true case of suboptimal introspection. So now, instead of asking people to rate their confidence in a way that relies on that internal criterion, we go to our friend's signal detection theory, and we use a two-interval forced choice. So in this type of task, you ask observers to make judgments about two stimuli, one after the other, and then to say which of the judgments they feel more confident in. So you show a participant a stimulus, and you ask them to make a first decision, and then you show them a second similar stimulus, and you ask them to make a second decision, and then you say, which one will you bet that you got right? Which one are you more confident in? So this completely removes any reliance on any internal criterion. So to use this type of two-interval force choice to test for blind sight-like suboptimal introspection, what we do is we actually remove the target in one of these intervals. So the idea here is if you can discriminate the target when it is there, but doing this discrimination task is subjectively or introspectively no different from discriminating nothing at all, like when the target isn't there, then what you'll do is you'll answer 50-50 when we ask you which decision you feel more confident in. So now you can do the task, but you're 50-50 on this confident judgment, betting on the target present or the target absent interval. So we arranged each trial in our experiment like this, where we mask an oriented Gabor patch target, so that's a sinusoidal grating, and it's there in the red square and it's tilted to the left or right of vertical. And we mask it with a forward and backward masking paradigm so that we present it sandwiched in time in very quick succession with these other colorful images. And in a second interval, we show the exact same thing, but the target now is not there. And we show people these two intervals at varying contrast levels for the target when it is there, up to 13 levels. And we ask them to bet on which decision they will get right. That is, which one they feel more confident in saying whether it was tilted to the left or right from vertical, and then also to indicate the orientation of that Gabor patch for both left and right tilted. And with this paradigm, we have a really easy way of detecting that blind sight-like suboptimality. So to do this, we plot the percent correct on the orientation task when the target is present on the x-axis. And on the y-axis, we plot the percent of time that participants say that the target present interval is the one that they're more confident in. And we would expect suboptimality of this blind sight type to create a curve that looks something like this. So here, where the curve is flat, right here, you can do the orientation discrimination task when the target is present. But doing this task is introspectively no different from doing the task based on an invisible target. So this would be the suboptimal introspection that we're looking for. And once we find that, then we can go about using various Bayesian observers to figure out why and how people are suboptimal. Seems like a good plan. But see, there's this pesky thing called data. And data get in the way when you have the very best of intentions. So we're looking for this elbow type behavior where it's flat and then it rises along as you get better on the orientation percent correct task. But we saw no trace of this elbow, this suboptimal behavior. We even did a second experiment where we played with feedback and the order of the questions, and the results look basically identical. And we did another experiment using continuous flash suppression, which is a different kind of masking. So instead of forward-backward masking, now we show the image to one eye, the target to one eye, and then we show to the other eye a lot of colorful images flashed in very quick succession. And we found the same behavior, and this work was done by an excellent graduate student in the lab by the name of J.D. Knotts. So it turns out, excuse me, that when you build a Bayesian ideal observer that does this orientation discrimination task when the target is there, and then it bets on which of these two intervals has a higher posterior probability, so that's the higher probability of it being correct in the orientation discrimination task. When you build an ideal observer to do this, you get essentially the same behavior. So this was pretty surprising to me. I really thought that I would be able to see this suboptimal behavior by using these techniques because that's what had been reported in many studies in the literature. And not only that, with this very conservative two-interval force choice paradigm, people don't even show a hint at suboptimality. They are ideal observers. So this is a very hard thing to reconcile with those blind sight-like studies that use visual masking that have been reported previously. But then I thought, well, maybe visual masking, this forward-backward masking or continuous flash suppression, maybe this is just not the right way to go about getting this behavior. Maybe we need to do something a little bit stronger than just manipulating the stimulus. So since I'd done some brain stimulation in the past, that seemed like a logical next step to try. So in collaboration with Tony Rowe at the Graduate Center of the City University of New York, we applied this two-interval forced choice confidence paradigm using transcranial magnetic stimulation, or TMS, to visual cortex at the back of your head. So this time we have all sorts of trials, so people didn't catch on to our trickery. So sometimes we put the target in both intervals, sometimes only in the TMS interval, and sometimes only in the no TMS interval. And of course we counterbalance all of the order. And so to get at those something versus nothing trials like we had in the previous experiment, we can look at these trials here, where we zap you when there is a target, and then we don't zap you when there is not a target. So this is akin to the something versus nothing kind of experiments we did previously. And to remind you, it's this elbow behavior where it's flat along the x-axis and then it rises as your orientation percent correct grows. This is the behavior we're looking for. But this is what we found. So you'd think that TMS to visual cortex would reduce confidence in your discrimination performance, but instead it increased it. When we look at those something versus nothing trials, people bet on that interval that contains the target and it gets TMS'd more often. Even when doing the task due to TMS, they're totally guessing at the orientation of the target. So here, when D prime is 0, that means they're totally guessing. So it's as if TMS artificially inflated the sense of confidence that people have in their discriminations, even as it really messed up their ability to do the discrimination to begin with. So this certainly isn't that suboptimal blindsight-like elbow behavior we were looking for, but it's also not ideal observer behavior either. So now we have to figure out what's going on here. So here's our theory about why we got this subjective inflation of confidence due to TMS. We can think about what the perceptual system is doing as taking samples from the environment. And these samples are corrupted by noise, which is environmental noise or internal processing noise. And this means that they form a distribution of evidence that you get through your eyes. So if you're asked to determine if something on a computer screen has a left tilt or a right tilt, the two relevant distributions would be these, left and right. So when the thing out there in the world has a left tilt, you will have some noise associated with your sensory experiences, such that you will sample on a trial-by-trial basis a distribution of evidence. And when the thing has a right tilt, you'll get other samples with other noise. And so now what we can do is we can put a criterion line in the middle and say that evidence that falls on one side of the criterion indicates a left tilt, and samples that fall on the other side of the criterion indicate a right-tilted thing out there in the world. So when you decide this thing has a right tilt, what you're saying is that the evidence you got on this particular trial probably came from this distribution on the right, here. And we can separate this evidence further into categories like strong evidence or weak evidence. So here, the strong evidence is far away from the decision criterion that separates left tilt from right tilt. And the weak evidence is much closer, so it might be mistaken for left tilt. So now that we've set up this signal processing environment, we can ask a very important question about what happens when we zap you with TMS. So TMS is likely going to cause a very substantial increase in the noise in your samples, right? It's going to make everything noisier, makes you worse at the task. So suddenly all of your samples are noisier. So when we draw the distributions, now they're going to be fatter, like this. So now your ability to tell whether something is tilted left or right is not as good. You're making more mistakes. So sometimes when a sample comes from the right tilt distribution, now you say left because it fell on the left side of the criterion. And sometimes when it comes from the left tilt distribution, you mistakenly say right. So this all makes sense. So Now, what does this increased noise do to our confidence judgments? Well, there's two possibilities. The first possibility is that you, the observer, have some way of measuring the noise in your own system. So the noise went up, and you know that it went up, you, the observer. And you, the observer, also know that you're making more mistakes as a result. You know that the information you're getting is less reliable because it's noisier. So what should this do to your confidence judgments? Well, they should go down, right? You have less confidence overall, and you know you're making more mistakes. The second possibility is kind of the opposite. You don't know that the noise went up because you have no way of monitoring the change in your system. So in this sense, you would be metacognitively blind to the increase in noise. So now you're making more mistakes, but you don't know that you're making more mistakes. And not only that, if you knew the noise increased, you should also update what counts as strong evidence. You should move this line here, this criterion for what counts as strong evidence, up to account for the increase in noise. But since you don't know the noise increased, you don't update where you put this line. And so you might say, hey, I actually have more of this strong evidence, this evidence that fell in the strong category. So your confidence will actually go up. So this second option is kind of counterintuitive, but it turns out that if you build a Bayesian observer model that implements this type of ignore the noise rule, that doesn't update what it counts as strong evidence, even when there is TMS noise, that it can explain this TMS result quite nicely. So here, this observer is metacognitively blind, and you see that the model does a very good job of capturing the data. Here, this Bayesian observer is metacognitively aware. It knew the noise increased, so it decreased its confidence judgments as a result. But this does not match the human participant's behavior on this task. Okay. So now some of you are probably saying to yourselves, wait a minute, when the noise increased, there was more of this weak evidence here. There's more of this too. And in fact, there was more of it even over on this side of the criterion. So everything should balance out in the confidence judgments. Even if you don't know, the noise went up, right? Well, there might be a second thing going on too that compounds this effect. Based on some previous psychophysics work, it seems like what happens is once you've decided, say, something is right tilt, when you go to judge confidence, all you care about is how right tilted it looks. So if we categorize the evidence you're getting as incongruent with your decision on the left or congruent with your decision on the right, at the same time as you're happily accepting all of this new strong evidence in favor of your decision as proof that you made the right choice, you're actually ignoring any evidence that is incongruent with your decision. This is the evidence that might indicate you made a wrong decision, but you just ignore it. So both of these things are going on. You don't change what you count as strong evidence, so you think there is more strong evidence, and you ignore that stuff over there that is incongruent with your decision. Now that you've decided the thing is right tilted, any evidence that it was left tilted is not relevant anymore. So now the evidence you do have, this decision congruent evidence here overall, has a higher mean. Your confidence in your decision goes up, even as your ability to do the task goes down. So the next question is, do we need to externally modulate the brain in order to see this kind of decision congruent evidence influence or rule for computing confidence in a decision? It turns out you don't even need to do brain stimulation to see that this is how the brain seems to compute confidence. You can do this with straight old psychophysics, basic behavioral tasks. So to test this theory, I designed a new study using just stimulus manipulation. So here, the basic idea is that you create two conditions that have matched performance but different confidence levels. So here we have a large tilt or low contrast condition, and here we have a small tilt or high contrast condition, and high contrast condition. And the task in both cases is to say whether this Gabor patch, this sinusoidal grating on the computer screen, is tilted left or right from vertical, just as before. But in one case, it's tilted 45 degrees, and in the other case, it's only tilted 5 degrees from vertical. And what we'll do is we will titrate the contrast levels in these two conditions such that performance on the left-right discrimination task is matched. It's equivalent across the two. So according to that decision-congruent evidence theory, what will happen is that because there will be more decision-congruent evidence in this condition, the high contrast small tilt condition, we should also see higher confidence in this case, even though performance is matched across the two conditions. So when we do the two interval force choice task with these two conditions and we plot our results the same way as before, not only do we not see that elbow that we were looking for originally, but now we see that people are indeed more willing to bet that they got the discrimination decision correct for the small tilt, high contrast interval than the large tilt, low contrast interval, even though, again, their performance is the same across the two. So the confidence judgment tags along with the amount of evidence you have favoring the decision that you made, which we manipulated here with the contrast. So it seems that we've got some really intriguing evidence that once you've made a decision about the identity of something you see out there in the world, like this cat, if you have to judge your confidence about whether you are right, all you care about is how much it looks like the thing that you decided it was, a cat. Once you've decided it's a cat, you really don't care how much it might also slightly resemble a dog or a fish or a bird in some other small way. You ignore all that other evidence that's incongruent with your decision and just report how cat-like this thing looks. So this is the theory. And there's some other evidence that this might be happening too. We see self-consistency effects throughout the literature. We see this font size effect. And we see even hints of this computation in higher cognitive computations such as confirmation bias or the representativeness heuristic. Okay, so if we think that this is what's actually happening in the brain, we need to test this theory with multiple methods because we are good scientists. Okay, so to look for neural evidence of this theory, we capitalized on a rare opportunity in human neuroscience to directly record signals from the surface of the brain using electrocorticography in epilepsy patients. So these patients are undergoing surgery for reasons other than this research, but we can use this opportunity to record what their brains are doing as they're doing some task. So we record signals from the surface of the brain while the subjects do this classic perceptual decision-making task. So here we show a stimulus, it's a degraded, noisy face or house stimulus, and we ask subjects to say on every trial, is this thing that we flash very briefly a face or a house, and how confident are you in your judgments? So we then use a wavelet transform to get these electrode signals into the time-frequency domain, and we see that the relevant signal is really focused in this high gamma range between about 80 to 120 hertz, and this is consistent with many previous reports of the task relevance of this frequency range. So this is where we focus our analysis. We take these time-frequency signals and we assign the power in the high gamma frequency bands as features to pass to a cross-validated linear support vector machine classifier. So this is machine learning now, to decode two behavioral factors on a trial-by-trial basis. So we train one classifier, one machine learning classifier, to decode what decision each participant made on every trial, a face or a house. And we train a second classifier to decode whether the participant indicated high or low confidence on a trial-by-trial basis. And we plot the decoding accuracy as a function of time after stimulus onset. And you can see that both behavioral factors can be decoded well above chance, beginning about 200 to 400 milliseconds after the onset of the stimulus. So the information is there in the brain, despite admitted limitations of ECOG spatial coverage and other factors. And we can see this face evidence and this house evidence there in the brain on a trial-by-trial basis, and we can see that it varies independently. So some trials have more both face and house evidence, and some have less face and house evidence, and the balance of the face and the house evidence changes on every trial. So next, now that we can get this face evidence and house evidence out of the brain using our support vector machine classifiers, now we can ask, how does this evidence get used to make confidence judgments? Can we find support for the hypothesis that people are actually ignoring that decision incongruent evidence when they judge confidence? So to answer this question, we take the trial-by-trial face evidence and house evidence, it varies independently, and we see what happens if we try to predict whether the person said face or house and low or high confidence on every trial using two possible rules. One rule that does not discard the decision incongruent evidence, and the other that does. So let's use these rules with an example trial to see how they work. On this first trial, you said face. There was more face evidence than house evidence, and you'd get it correct. And the first rule that we examine is what we call the balance of evidence, or the difference between the decision congruent and the decision incongruent evidence. So you're using both. So you chose face on this trial. So this balance of evidence is going to be how much face evidence you had minus how much house evidence you had. Pretty straightforward. We're also going to try a second rule for predicting our subject's behavior, which is the magnitude of the decision congruent evidence alone, regardless of the decision incongruent evidence, as if the observer ignores that incongruent evidence. So on this trial, you chose face, and you have some level of face evidence, so we'll see whether that by itself, without taking into account the house evidence, is also predictive of your behavior. And we'll check how well these two rules are able to predict our subject's decisions about face versus house on every trial, and also how well they're able to predict their confidence judgments. And so this is called a choice probability analysis. And what we see now is that we're much better able to predict our subjects' face versus house decisions, the decisions, if we take the balance of face and house evidence together rather than just looking at one. So now we're looking at the left side of the graph here, and you can see that the balance of evidence rule really does much better at predicting whether someone says face or house on every trial than if you just look at the evidence that is congruent with their decision. But this pattern is very different for the confidence judgments. So we didn't actually gain anything at all by taking the evidence favoring the decision the observer did not make into account. It's as if the amount of that decision incongruent evidence is totally irrelevant for the confidence judgments, like they're ignoring it altogether, just like we saw in the psychophysics and TMS results earlier. So now that we've seen some evidence that this strategy might be what the brain is doing in calculating confidence, we should probably see if it's even biologically plausible for a brain to do this at all. So to see what I mean, let's build a little model. So here we have units that each accumulate evidence for a particular stimulus category. These kinds of units here essentially add up the evidence for one type of stimulus over time. And these are like neurons in areas such as LIP or FEF or superior colliculus. So in our toy example, this unit on the left, likes vertical things, and this unit on the right likes horizontal things. And this mini network is set up with a strong degree of lateral inhibition between these two units that have opposing tuning preferences, such that the more one guy fires, the more he inhibits or suppresses the other guy. And this strong lateral inhibition is actually a really great thing for neurons to do, because it helps the network not be susceptible to random fluctuations that don't have anything to do with the stimulus that's out there in the world. It's really able to average out the noise very well. And importantly, when we have strong lateral inhibition like this, we get out of this network the balance of the evidence between the evidence for vertical and the evidence for horizontal. So this is akin to the posterior probability in a Bayesian sense. It takes into account both the decision congruent evidence for the choice that you'll ultimately make and the decision incongruent evidence for the other possible choice by subtracting off that decision incongruent evidence from the overall estimate at every time step. So let's say I show this little toy network a stimulus that's a blend between a vertical and a horizontal stimulus, and I ask it to make a decision. Is this stimulus more vertical or more horizontal? And the units start accumulating evidence, and the more the vertical one fires, the more he inhibits the other guy. So the output of this network is something like the difference in firing rate for the vertical unit versus the horizontal unit. And so at the time of the decision, which is here when it reaches a decision boundary, the difference in firing rates for the vertical versus the horizontal unit will look something like this. And if I show this network a similar stimulus that has the same balance of verticalness and horizontalness, but I turn up the overall contrast, I'll get the same accumulation of relative evidence here, and at the time of the decision, here, I will get the same firing rate ratio at the end. So we'll get the same discrimination performance out of these two conditions, as we showed earlier with our psychophysics and as other researchers have shown as well. But remember, this balance of the decision congruent, subtracting off the decision incongruent evidence, this can't be the only thing that's retained by the system, because we know that there are different confidence judgments associated with these two conditions. This one, the low evidence condition, will have lower confidence than this one, the high evidence condition. So somehow we have to be able to keep track of the absolute evidence for vertical and horizontal without doing that subtraction. So what we'll do is we'll modify this perfectly normalized network so now we can assign how much the neurons inhibit each other to be this parameter W, such that when W is big, the units really inhibit each other, and we get this perfectly normalized network, which is like a drift diffusion kind of network. And when, in contrast, we have small lateral inhibition, we have a race model. So we can also store the absolute amount of evidence that existed for each of these choice alternatives. So here, this is the balance of evidence, and here, the units accumulate evidence independently and don't really care what each other are doing. Now, most previous work has assumed that one or the other of these types of normalization or lateral inhibition is occurring in the brain. And there's certainly evidence that each model type describes behavior well under different circumstances. But I don't think that we have to choose just one. I think that we can have the best of both worlds, because the truth is that all different levels of this lateral inhibition exist in the brain. Some units are more normalized or more laterally inhibited, and some are less. And this is called tuned normalization, and it's now been reported by several groups doing recordings of rhesus macaque or monkey cortex. And just to put one more thing in the backs of your brains as we go forward, I'd like you to think about what these neuron types might be particularly good for in the sense of tasks that an organism has to solve in the real world. So perhaps because they're better at averaging out the noise, these diffusion units are better at discrimination type tasks, fine-grained discrimination. But in contrast, the unnormalized units that have a small lateral inhibition, they're less inhibited overall, and so they're better at detecting things out there in the world due to less inhibition. And these tasks are both important for an organism to be able to do. So let's now see how this tuned normalization might be built into an actual network of neurons. So now we'll start with this network that we had before, and we can assume that that lateral inhibition is accomplished by inhibitory interneurons. So that's this neuron here and this neuron here. And we explicitly specify the recurrent excitation and the leak that these units have such that they look a little bit more like biologically plausible neurons. And our next step now is to create two populations of these units, a more inhibited or more normalized set where the degree of lateral inhibition is high, and a less normalized set where the degree of lateral inhibition is low. So now we can do some cleanup because we can assume that units that have the same tuning preferences don't have separate pools of inhibitory interneurons. So this unit and this unit like vertical things, and they excite the same inhibitory interneuron that then inhibits the guys that have opposing tuning preferences. And we can imagine that the equivalent connections go on over here for those that like horizontal, and now we can parameterize. And we can say that how each unit updates its firing rate activity on a time-step by time-step basis is a function of this scaled input drive, subtract off some leak, add on some recurrent excitation, but then here we want to subtract off the amount of firing that opposing units have, units that have opposing tuning preferences, and we scale that subtraction by whether a unit is more or less normalized overall. So that's nice. Now we have 4 units instead of 2. This isn't much of a network though, and it doesn't really explain how tuned normalization relates to decisions or confidence judgments quite yet. So to do that, we should make our network bigger. We need more than just 4 units, so now we have our inputs vertical and horizontal, and now they're connected to a layer of these accumulator units where we have a lot of these units, and each of these accumulator units has the same recurrent excitation and leak of our previous little toy version. And now we add recurrent excitation among similarly tuned units, such that units that have the same tuning preferences excite each other based on how similar their preferences are, and lateral inhibition based on the same idea. Units that are opposingly tuned inhibit each other, and units that have the same tuning don't inhibit each other. And then what we'll do is we'll scale this lateral inhibition based on whether we're assigning the unit to be more normalized or less normalized. So now we can finally hypothesize about how normalization may relate to decisions versus confidence judgments. So we tried out some different models, and it turns out that the one that does a great job of explaining some surprising findings in the literature is one that works like this, where the more normalized, or what we called those discrimination units, contribute more to the decisions, and the less normalized, or what we called the detection units, contribute more to the confidence judgments. And once you've made a decision, say horizontal, then confidence is read out as the decision-congruent unit's activity based on how normalized they are. Okay, so the next big question is, how does this network do? Can we actually predict behavior using this neural network? And it turns out that we can. It actually gets everything right. So it captures that you can have matched performance but different confidence. That when you add noise with TMS, you'll get higher confidence rather than lower confidence. It captures that confidence and reaction time are inversely correlated, which lots of people have shown. And it also captures some counterintuitive effects that have been reported in the literature. Like when you do some very clever and bizarre stimulus manipulations, you can change metacognitive sensitivity. So that's the correlation between confidence and accuracy on every trial. You can make this go down even as people get better at the task, even as their accuracy is going up. And you can also introduce not noise, but volatility into the stimulus and not change accuracy. So accuracy stays the same, but confidence goes up. And this model predicts all of these very strange behavioral reports. So we've now been working with Michelle Basso's group here at UCLA to try to identify tuned normalization in the superior colliculus of rhesus macaques, monkeys, using multi-channel electrodes. And so far, we do see some evidence that tuned normalization does exist in this oculomotor decision-making area. And this lends further credence to the hypothesis that this neuronal property is directly related to the perceptual decision-making apparatus. So we're currently working on critically examining the evidence that we found and relating this tuned normalization to choice and confidence behavior as produced by these monkeys. So what this really says to me is, we have this circuitry that's kind of specialized for fine-grained discrimination on the one hand, and where where you can average out the noise. And then on the other hand, you've got these units that can detect minute amounts of information out there in the environment. And both of these abilities are really important for an organism to have. And it seems like it is this detection half of the circuitry that's primarily recruited when you ask an observer to make introspective computations, like judging confidence in a decision. Okay, so really all this can be summed up as you're doing some task. Once you've made a choice, to report your confidence in that choice, all you care about is how much evidence there was favoring your decision. Once that decision is made, any evidence that could have supported an opposing interpretation is tossed out. It's no longer relevant to subsequent introspective evaluations. And we've seen this across a number of studies now from our lab and other labs too. And it seems like it extends beyond perceptual decisions, even to fear detection. And it turns out that this strategy for introspective confidence is not even unique to humans. So right now, like I said, I'm doing some really exciting follow-up work with Michelle Basso in non-human primates. And I'm also working with Alicia Izquierdo here at UCLA to do a similar type of task in rodents. And it turns out that even rats show this same behavioral effect. So here, we've matched performance on these two conditions, but on the one that has higher evidence, the rats wait longer for a reward, showing that they are more confident. So the great thing about working with animals other than humans is that you can really capitalize on exciting techniques that are unavailable in human work. So while human neuroscience is great, I think it's really important to capitalize on these exciting technical advances that are possible in animal research. Okay. So, decision-congruent confidence. It seems kind of silly when you think about it, that the brain is actually tossing out information that you'd think would be relevant to its decision and its confidence judgments. But yes, this is in fact what people, monkeys, and rats seem to be doing. And yes, this does seem to be what's happening in the brain too, that the brain has access to that decision incongruent information. We can see it. We can use it to predict a face versus a house decision, but then when it comes to confidence judgments, the brain totally ignores it. We also saw that this strategy, however suboptimal it may seem, is actually biologically plausible, and it produces all of these kinds of funny confidence behaviors. So now, I have to take a step back and say, okay, I started this whole talk by talking about consciousness, but then I tricked you, and I spent the whole time telling you about confidence. But really, I think we can learn a lot about consciousness from studying confidence, and here's why. I think the two are inextricably intertwined. What kind of conscious perception do you have if you have zero confidence? What would it mean to say that you actually see something, but you have no confidence, not low confidence, but zero confidence in what you're seeing? Think about it. If your perception is genuinely conscious, it should always come with at least some level of confidence. It doesn't make sense to see something and have zero confidence in what you're seeing. So this seems to imply that confidence is necessary for awareness, that whenever there is even a little awareness, confidence comes along for the ride. So I've argued that confidence is necessary for consciousness, but is it sufficient? For example, it's conceivable that you might have confidence about something that you've decided without being conscious of the thing itself or of your decision about it. So you might argue that in that first study I talked about, that very first study where you have to discriminate two things and bet on which one you're more likely to get right, that perhaps what people were doing in this confidence betting task was based on some sort of unconscious hunch. They didn't see the stimulus, but they just had a gut feeling that they were going to get it right more often in that target present interval. So they bet based on that gut feeling. Well, what I didn't show you, though, is that if we change the question and we ask people to simply indicate which target was more visible, regardless of whether they think they got it right. They show essentially the same behavior. There's no blind sight when you change the question either. So this means that when people are confident in something, even just a little, they are conscious of it, even just a little, and vice versa. So in humans, at least, who have no neurological deficits, we have empirical evidence that confidence is a sufficient indicator for consciousness as well. So the point is that these two things, confidence and consciousness, they're really very connected. And they're connected because the circuitry underlying detection of an object is what gets recruited for metacognitive or introspective judgments of certainty in that decision. Confidence and consciousness are interconnected because they rely on essentially the same computational machinery. And because the introspective system is set up this way, you get these funny behaviors. Like when something that looks a lot like what you decided it is, you're very confident in your decision about its identity because you discount any aspects of it that might suggest it's something else. And this happens especially when the reason it looks a lot like what you decided it is because you've got a lot of noise in your system, like when we zapped you with TMS and you have more noise than you think you do. So this can happen when we zap you with TMS. So we introduce a bunch of noise, but it could also happen that the perceptual system underestimates the amount of noise that it has for other reasons. So one example of this that I really like is in the periphery of our vision. So our rods at the edge of our retinas are very bad at detecting fine detail, and they're completely unable to detect color. Yet what we subjectively experience in the periphery is a whole lot more like this picture on the right, than the picture on the left. We don't think we see only black and white and fuzzy things in our visual periphery. No, instead we see color. We see fine detail, or we think we do. We actually have an inflated sense of confidence in the color and the detail that we've perceived. And maybe the reason is that the system has underestimated the noise that occurs in the periphery of our vision. So others in the lab now are investigating this type of subjective inflation in the periphery. So this decision-congruent evidence model really does show how errors in the calculation of confidence, an over-reliance on decision-congruent evidence, that this can really account for the nature of subjective experience or awareness itself. So with that, I'd like to thank all of my collaborators without whom this work would not have been possible, and also our funding sources, and you all, my audience, for your attention. Thank you.
Thank you, Dr. Peters, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. And Dr. Peters will answer as many questions as we have time for today. So without hesitation, let's jump in to the questions. Excellent. Dr. Peters, when you decoded decisions and confidence, did the location of the information change in the brain?
This is a really good question, is how the representations of decision versus confidence, do they look the same even though the calculations look like they're different? So I think the best way to answer this question is to show you some work that we did on trying to get at what those representations look like in the brain and where they are and when they arise. So here on the upper left of the graph is what I showed earlier in the presentation. But what we can do from this decoding of the decisions and the confidence judgments is we can actually parcellate the information so that you can assign to every electrode that we record from across all of our participants how much each electrode at each spot in the brain contributes to our ability to decode. So to the extent that we can actually decode decisions or confidence judgments, which electrodes are the important ones to being able to do that decoding? So now if we look at the lower part of the graph here, panel B, you can see these are the little dots represent all of the electrodes that we have recorded from all of our subjects. And you can see that on the top row of panel B, that the decision information really does seem to rely more on these occipital electrodes here, especially between 200 to 600 milliseconds after the stimulus onset. And the representation looks a little bit different for the confidence judgments. There are fewer red dots overall, but there are some more red dots that appear here, more in like the parietal and even frontal regions. And so to get at this distribution of information in a slightly different way, the next thing that we did was we can group these electrodes by whether they belong to one of the 4 neocortical lobes. So the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. And then we can plot the average contribution of electrodes that are in that neocortical lobe as a function of time after the stimulus onset. And so that's what's plotted up here in panel C. And so you can see that when we split it up by the neocortical lobe, that for decisions, it's really the occipital electrodes that are doing the heavy lifting. And to a little bit of extent, it's the parietal electrodes too, the temporal electrodes, but those frontal and parietal electrodes, especially early in the trial period, are not actually doing as much of the heavy lifting. They do tend to be more important later. And this pattern is really quite different for the confidence judgments. So you do see this little occipital spike here, but it's later than the occipital spike that you see for the decisions, and it's not as strong, and it's not as sustained. And then we also see then that despite the fact that during decisions, occipital electrodes are really winning, they're contributing the most for most of the peristimulus window, that here it actually switches for the confidence judgments such that first it's the occipital electrodes and then it's the temporal electrodes that are doing the heavy lifting. So this shows us that even though we can decode the representations to a similar extent for both decisions and confidence judgments, that the nature of these representations is really quite different. And one more piece of information that I can show you that the representations are really very different from each other is here. So now that we know how much each electrode contributes to decoding each behavioral factor, we can see which are the most important electrodes for decoding decision and confidence judgments. And then we can see where those most important electrodes are and see where they overlap. And so to the extent that you do have electrodes that significantly contribute to both decisions and confidence judgments, they're pretty much in the occipital region, maybe a little bit in the temporal region. But if you look at the y-axis over here, you can see that there's really not a lot of feature overlap. So the electrodes that are important for decoding decisions are really not the same as the electrodes and the features that are important for decoding confidence judgments. So I hope that answers your question.
Excellent. Thank you so much for that answer. Our second question of the day asks, in the blind sight study, maybe people had a gut feeling or a hunch, but still didn't actually see the target, and that's what made them able to do the betting test and not show blind sight.
Right. So this question I actually get a lot when I talk about this study. Part of it can be answered by the similarity between the answers that participants gave when we asked them to report their confidence versus when they reported just the visibility of the target. So you'd think that if they're doing the confidence judgment on the basis of some gut feeling or hunch, that maybe they wouldn't be depending on the same thing when we ask them about visibility, that instead, hopefully what they're reporting is the actual visual experience or visual qualia of those stimuli. But I think this also can really be answered by the way that we define blind sight, the way that we define subjective experience in this task. So what we're asking with this task is, here, if I go to this slide, what we're asking with this task is, if you can do this task above chance when the target is actually there, but doing so is introspectively or subjectively no different from doing the task when the target is physically not there, then you will be at 50-50. And so having a gut feeling or hunch, in essence, there is something that it is like, to use philosophical language, something that it's like to have that gut feeling or hunch that is subjectively different from doing the task based on nothing. And so in that way, we would say that these people are not actually metacognitively blind. They do have some introspective ability to do the task on the basis of that gut feeling or hunch, even if it's not visual in nature.
Thank you, Dr. Peters. Our next question of the day asks, how do you measure tuned normalization in the monkey's brains?
This is a great question. So to answer this question, I'll show you the stimuli that we use. So we actually made use of some data that had been collected previously that were not having to do with this study, but they turned out to be very useful. So in the type of task that we use, the monkey is fixating on the screen, and then, so here, and we record from the neurons in the superior colliculus using a multi-channel V probe. And so previous to the task, you actually map what's called the response field of these superior colliculus neurons. So this is the area in the visual field that these neurons really like. And then a dot motion stimulus comes on, and then you measure the way that the neurons respond to this dot motion stimulus. And the stimuli that we were interested in were cases where you've got a bunch of dots moving in one direction, or you've got a bunch of dots moving in two directions. And it's the same percentage of dots moving in one direction or the other. So we can call this preferred, 100% preferred, or 50-50 preferred and non-preferred stimuli. And so then we can measure how these neurons fire in response to these stimuli. And so in panel C, you can see that when it's 100% what we call the preferred stimulus, so this indicates that the monkey should make a choice towards that response field, that the neurons really like that. They fire a lot more than when it's 50-50 preferred and non-preferred. So for each neuron then, we can calculate how much they change their firing rate when there's only preferred stimuli on the screen versus when there's both preferred and non-preferred stimuli on the screen. And every neuron changes its firing rate a little bit differently. And from that difference, we calculate a modulation index. So the higher the modulation index, the more normalized the neuron is, the more it's inhibited by other neurons that have opposing tuning preferences. And the smaller the normalization index, the less normalized the unit is. The closer to 0 it is, the less normalized it is. And when we calculate this modulation index on the basis of disjoint trial subsets, so we just split the trials into even and odd trials, and we calculate this modulation index separately, we see a really nice correlation between the even and odd trials. So this shows that tuned normalization really does exist in the superior colliculus. And this is actually the first report of tuned normalization outside the cortex in a subcortical structure, and it's the first report of tuned normalization in these accumulation type units. So we're really excited about that.
Okay, well, thank you so much, Dr. Peters, for your time today. We truly do appreciate it, and that is all the time we have for questions. Do you have any final comments that you would like to close today with?
Really, I'd just like to thank everybody for your attention, for listening to me talk about my research. It's very exciting to be here, and thank you very much to LabRoots also for putting this together and inviting me to participate.
Well, and it's been our pleasure to have you present. I'd like to thank the audience for joining us today and for their interesting questions. We would like to thank LabRoots for today's educational webcast. This webcast can be viewed on demand through June 2017. LabRoots will alert you via email when it is available for replay. We encourage you to share that email with your colleagues who may have missed today's live events. I am Christina Mahalik with LabRoots, and we'll see you here next time at labroots.com. Goodbye.